Thanks again, everybody, for joining us. I think we've all found that Zoom is such a great way to stay in touch while we're all hanging out at home. And I want to thank you, Professor Adrian, for joining us. And I'm going to do just a quick little intro. So Professor Bird received his PhD in biochemistry from the University of Edinburgh. He did a postdoc at both Yale University and the University of Zurich. And then he returned to his alma mater in 1990 as Buchanan Chair of Genetics, a position that he holds to this day. He's the recipient of many awards, too many to list, but I would like to congratulate him on the most recent one, the Brain Prize, which he shares with Dr. Huda Zogby. And I think a noteworthy honor is the fact that in 2014, he was knighted by Queen Elizabeth. Very impressive accomplishment. Now, in our own Rett world, Professor Bird has made a number of very remarkable contributions. He discovered the MECP2 protein about 30 years ago. He made the very first animal model of the disorder, an animal model that's been used in hundreds of laboratories since then, which was, I believe, 2001. In 2007, he published a landmark paper announcing that the symptoms of Rett syndrome are reversible, remarkably and unexpectedly reversible, at least in mice. And he's also helped to reshape the scientific thinking about the function of MECP2. I first connected with Professor Bird. I remember the exact day, October 21st, 1999, because I keep a log of most of my calls. And here are the notes from that very first call that I had. And, you know, we've been working together and communicating ever since then. He's a trustee at the Rett Syndrome Research Trust. And we've appreciated his advice and his guidance all these many years. So thank you so much, Professor Bird, for joining us today.
Great pleasure.
Just housekeeping comments, since more people have joined. The participant's video is off and the microphones are muted, but you will have an opportunity to ask questions via the Q&A. If you have I'd like to wait for most of the Q&A questions until the end, but if you have a burning question, go ahead and ask it, and I will keep an eye and try my best to facilitate the questions as we go along. So I'm going to just stop sharing the screen right now. Okay. And before we dig into the science of it all, I thought we'd start with a more lighthearted question and ask you what you like to do in your free time. What do you consider fun outside of the lab?
Well, I quite like, I like photographing animals, really. I would say birds. I like just wandering around. I don't really sit in hides and wait for them for hours and hours, but I like wandering around with a camera, and these days it's a lot easier in just photographing them. I like cooking, which is kind of, it's all the science I get to do these days. Only the good aspect is you get to eat it afterwards. And I actually like eating as well, which is a good thing. I have a total of 4 kids, so that also occupies a certain amount of time, though fortunately a lot less than it did because they're quite old.
Okay, and you're also quite the athlete, I would say, over the years, right?
I used to play field, what you call field hockey, and cricket. Not quintessentially American games, but nevertheless, I enjoyed those. And I played a bit of tennis, and I was into hill running, running up and down hills for pleasure, which to many people seems more like purgatory than pleasure. But my knees, unfortunately, aren't up to it anymore.
So one of the questions that I've been getting a lot for the last several months, as you might imagine, is how is the pandemic affecting research? Is it slowing things down? And, most of the labs, if not all of the labs that are doing Rett research that RSRT is funding, have been closed down. Some of the companies are continuing to do their work, like Jackson Labs, for example, but the academic labs are shut down. Do you want to say a little bit about how the pandemic has kind of affected your lab in particular? What are you able to do and what have you had to put on hold?
Yeah, we've been off now for quite a few weeks. We can't do anything, no experiments. We've fortunately got our mice, which are critical for our work because we use them as a model for Rett syndrome. Fortunately, we've been able to keep reasonable numbers of them, but it's going to take us a little while to ramp up once we get in. We're basically not the only people who are allowed into our building are people who are working on COVID-19 directly, and we're not. So we can't go in, and the anticipated return is probably something like 2 to 3 weeks before they will open some buildings as a trial, apparently. I'm not sure what that trial is going to be about, but we're hoping to get in within the month, let's put it that way, and get back to work. It's frustrating. We talk a lot, a few times a week. We still have our lab meetings, etc. But nobody can do any experiments, which is difficult to take, but necessary.
Yeah. So what I'm hearing also from other investigators, just for information for our viewers, data is being analyzed. Things like bioinformatics analysis can happen. A lot of people are doing a lot of reading, a lot of thinking about future experiments.
Yeah.
Which
it's
is
true, actually. And you could argue that one spends, quite a lot of time doing things, rushing around, trying to get studies finished, papers out, etc. And it's not a bad thing, not a totally bad thing, to have time to reflect and discuss which way we should move forward. And we've been doing a bit of that. So it's, although it's not great and it's slowed us down a lot, it nevertheless has a good side.
Yes. One silver lining. Okay, so let's get started with our kind of science-based questions. So what drove you to start studying MECP2, or you actually didn't know it was MECP2 yet you were searching for it, but what drove you to look for this type of gene back in the 80s?
Well, I'm not a clinician. I'm a basic scientist, and I got into this because I was really interested in DNA and how it worked. And one aspect of how it worked was regulation of gene expression. I mean, the skin cell is different from a liver cell. It's different from a muscle cell, and they're different because they activate different genes. And genes make proteins, so the proteins in a muscle are different from the proteins in skin. Some of them are the same, but key ones are different. And so the question is how, given that the genome is more or less the same in every one of these cell types, how do you decide which genes you're going to switch on and which genes you're going to switch off? And it turns out that that's quite a complicated question to answer, but one way that we were interested in to begin with is the idea that actually the DNA gets stamped with a chemical mark called methylation, and that maybe differences in gene expression were influenced by where that methylation is put. And so we started off really by mapping where the methylation was. I mean, there weren't really ways of mapping where it was in the genome when I started out, but we contributed one of those key methods. And then the question was, okay, that's where it is, and it changes a bit when genes are switched on, but how does the whole thing work? How's it read? And it was really looking for proteins that might read that chemical marking on the genome that got us into looking for proteins that would bind to that. It was slightly more convoluted than that, but in the end, that's what we stumbled upon, a set of proteins that could bind to DNA when there were sites of this methylation on it, but couldn't bind to the same bit of DNA if you removed the methylation marks. And one of those proteins, one of the first ones we found, actually the second one we found, that's why it's got a 2 at the end, was MECP2.
What happened to MECP2-1?
MECP1.
MECP1.
MECP1. Well, I mean, in a way, the reason why we couldn't find MECP1, we started off looking for it, but we couldn't manage to purify it. It actually took us longer to purify. So someone else in the lab said, well, there's another one there. I'm going to go for that one. Turned out that one was much more abundant, particularly in brain. And so we were able to purify it from brain. And we sent it to people who could sequence bits of protein. And that allowed us to be able to go from having the protein to finding the gene that encoded that protein, the MECP2 gene. And from then on, it's much easier to study what proteins do. So the link, the connection between MECP2 and Rett syndrome was discovered in 1999, late 99, by Huda Zogby and her lab members. What was known about MECP2 before then? Yeah, well, we published the existence of MECP2 protein in 1992, and we said this is a protein that binds to sites of methylation on the genome, and therefore it might be involved in the regulation of gene expression and switching genes on and switching genes off. And we had established that it really did choose between methylated sites and non-methylated sites because there was an element of uncertainty as to whether or not what we found with the purified protein in the test tube is that what it actually did in a real living cell. We showed to our satisfaction that what it did in a living cell was that too. That wasn't the end of all the controversies because scientists, being what they are, quite rightly came up with alternative ideas that no, that is only one of the things it does. But we're now pretty sure that that really is its core function. It binds to methylated sites in the genome. And we also knew that it brought in a big molecular machine that silenced, turned down anyway, genes that were on. So if a gene was on, happily making protein, if it has this methylation on and then you add MECP2 brings in this big machine which says, whoa, slow down a bit. It's sort of downward pressure on the expression of genes. And that's actually what we hypothesized in the 1998. We published a paper in Nature in 1998 suggesting that. And in fact, that has turned out to be correct. There have been lots of meanderings backwards and forwards. No, maybe it does something different. But in the end, it's now that the evidence is really good, not just from our lab, but from several other labs, that its function really is that. So we had an inkling that that was its function at the time it was discovered. So that actually has, although it may seem to many people that this field goes very, very slowly, we seem to just know the same thing year after year. In fact, the study of MECP2 has moved quite quickly. And it was an advantage to know something about it when Huda's lab first implicated it in Rett syndrome. So that's a good segue into my next question, which is how did Dr. Zogby's discovery change the direction of your research in your own lab? Well, I could just mention to start with that at the time we discovered this protein, my lab was in Vienna. It was 1987, 1988, 99. My lab was based in Vienna at the Institute for Molecular Pathology. And the reason why I mention that is because Andreas Rett was alive and working in Vienna at that time, the very same time. But unfortunately, by 1999, he had passed away. And so I may have shared the tram with him going to the lab. and may have passed him in the street, may even have been at seminars with him, but I never spoke to him or acknowledged him or knew him, even though ultimately we were working on the same thing from opposite ends, if you like. So when Huda's paper was published, and your telephone call that you have the notes on was just after that, I had never heard of Rett syndrome. It turned out I had a few papers about it in my reprint collection I subsequently discovered, but I never really, it was reckoned to be a mutation, a gene that was caused by defective mitochondria, which are the sort of power packs in the cell. And that was what the papers I had suggested. So suddenly there was this protein that we'd been working on by then for 7 years, that was implicated in this neurological disorder that I didn't know anything about. And a parent, you, phoning me up, asking me what's going on. Huda Zogby also phoned me up, saying, we need to know more about this protein soon. It changed the direction of our research profoundly. We were already making a mouse model for blue skies reasons. We wanted blue skies research. We wanted to know how the protein worked. We didn't realize at that time that we were making the model that, as you say, has been used in hundreds of labs as a model of Rett syndrome. But of course, it didn't take us long to adapt to that. And I have to say, it was, although I prided myself before that almost on being a pure science person, indifferent to the world, of practicality, it's been an enormous benefit to our research to realize that actually what we were doing and what we have been doing since actually has relevance. Frustrating that it doesn't go faster, but nevertheless exciting, added an exciting dimension that has come to dominate my lab's work since.
Thank you. So as you look back on the last, let's say 2 decades, what do you think are some of the key discoveries that have taken place?
Well, the linking of the gene to the mutation, the MECP2 gene to the mutation, was obviously profoundly important, that Huda's lab did. It turned out to be a single gene disorder. It's most of the cases, not absolutely all the cases, but nearly all the cases are caused by mutations in the MECP2 gene. That was a key discovery. I would say that the discovery, the mouse model turns out to be an excellent model of Rett syndrome. It really does look as though what MECP2 does in the mouse is the same as what it does in humans. And that can't be said for all animal models of human disease, because mice are obviously not people, and certain things they do differently. There are bits of the brain that are very different, and some even absent in mice that are present in humans. So you've got to be careful. But everything that's been done suggests that it does the same in humans, and that's been vitally important, the model. And then, of course, the reversal was the reversal was a long shot. I mean, I didn't actually. I had some funding from RSRT, F as it was then, for this research, but I'd never really wrote a grant saying we want to restore the function of MECP2 in the brain, because it seemed a bit of a harebrained experiment, because everybody knew, in quotes, knew that brain disorders of neurodevelopmental diseases of this kind, by the time they had begun and become apparent, that it was too late to reverse them. And that was thought to be the case in mice as well. And that's why the study attracted so much attention, because not only us, but also the field was expecting the opposite result. We thought, yes, you can switch it back on, but tough, it's too late. And the answer we got was not just a little bit the opposite of that, it was completely the opposite of that. And it was really the tenacity of someone in my lab, Jackie Guy, that carried through that experiment and did all the beautiful work that led up to that result being so clear. So that was a bit of a shock, but we all know the implication that it had for studies of Rett syndrome generally.
Before you move on to the next thing, I might add just a couple of observations about the reversal experiment. I think one is the importance of, not assuming things in science, right? And just actually doing experiments to confirm or not what a hypothesis might be. And I think in a way, you as a non-neuroscientist, I think dove into that experiment maybe more so than a neuroscientist who might have come with a little bit more, dogma. I think it needed somebody like outside the neuroscience field to actually do that experiment.
I think that's to a certain extent true. It's rather like the questions that children ask are often the most profound questions because they don't come with any preconceived baggage. And our childlike, it's a nice idea that we ask that question in a childlike way, but I think I subsequently found that there were other labs who were also trying to do the same thing, and they were neuroscience labs. So it was not totally correct to say that we asked the unaskable question, though everybody was prepared for it not to be reversible, I have to say. So that was a surprise. And neuroscience, like every other field that I have been involved in, has its sets of beliefs in the way you say. If you think, for example, of for people who know about Dolly the Sheep, Dolly the Sheep was made from a cell from the mammary epithelium of another sheep, just taking one cell and turning it into an entire sheep. That was thought to be impossible because it was a differentiated cell. It had already decided what it was going to be. And the idea was, once you decided what sort of cell you were going to be, in this case, a mammary epithelium, then you couldn't be changed. And that turned out to be wrong. Same sort of thing in a way. Living things are much more plastic if you know the tricks for getting them to change than everybody had thought.
And I think the other observation, it was a three year in the making experiment, if I remember correctly, and there were some ups and downs. Like for a while, I remember getting a call from you saying things didn't look good, the mice were dying. As it turned out, it was because of the spacing of the tamoxifen injections.
That's right. That's right. We used tamoxifen, I should say, not as a hormone, but as a trigger to switch the gene back on because of sort of genetic engineering trick. And when Jackie gave it on 5 consecutive days by injection, some of the mice died, and it turned out that the reason was they didn't like being injected with tamoxifen. We think, to be honest, there's an element of uncertainty. It could have been that we were switching on the gene too quickly, and therefore there was an element of toxicity associated with that. I'm not sure I believe that based on what I've heard about how the experiments were done and what I've seen of the data, but certainly that was it. You're right, there were ups and downs, and when you see a mouse that was terminally ill suddenly start springing around the cage, your first instinct is that somebody's made a mistake, and that isn't actually the same mouse. Somebody's mislabeled it. So it took us a while to be able to believe what was found, but now of course it's been repeated in other labs and is secure.
Yeah, and I think those are important lessons for parents, myself included, right? That good science takes time, that sometimes you can't rush it. You actually, it takes time to do experiments. I get asked questions a lot about, preliminary results. What can you tell us now? Well, sometimes it's not good to share preliminary results because they are that preliminary, and you have to actually wait until experiments are done, checked and triple checked. And then even when something is published, to make sure that it can be independently confirmed in other labs.
Yes, I mean, you can see it now with the COVID. You know, people say, well, you know, we've known about this virus for 3 months. Well, why don't you have a vaccine yet? Why don't you have an antibody test? And all of these things take a while, and it's perfectly possible to believe you have a vaccine or an antibody test or some sort of drug that works, and then it turns out not to be the case when you do it more carefully. Biology is unfortunately rather messy and difficult to get definitive answers about. And it takes quite a while, and you have to not just do one animal that gets better, you have to do a number of them, and they all have to get better in the same way before you really believe you've got an answer. I think that's why scientists are always rather boring to interview, because they always say, well, we might have made progress. We're not really sure yet. It's going to take a while. They always seem to say that. But that's unfortunately the way science is.
So let's turn our attention to what parents are, always very interested to hear about, which are, potential strategies for therapeutics. And at RSRT, we have focused on strategies that target the root cause of the disease. We're lucky. I'll use lucky in quotations, lucky that Rett is a single gene disorder. We know the gene, it's not degenerative. You showed us that it's potentially reversible. And so these are all really good reasons why, we can think about going after the root cause of the disease. And there's lots of different ways that we can do that. The end result, the end goal is always the same, trying to boost levels of MECP2, but you can do that through gene therapy. You can do that through genome editing. You can do that through RNA strategies and, other ways. So knowing what you know about the various therapeutic strategies, what are your thoughts on potential, therapeutics that might be coming down the pike? Yeah,
I mean, obviously, Rett syndrome is caused by a mutation. It's caused by a mistake in the DNA. And DNA it looks like a twisted ladder, but each rung of the ladder is a bit of information, if you like. And Rett syndrome is caused by one of those bits of information or more being wrong. Because although DNA, when it's copied, is copied incredibly accurately, unbelievably accurately, there are mistakes every so often. And so 1 in 10,000 germ cells, sperm usually, or eggs sometimes, has a mistake in the MECP2 gene. So what you'd like to be able to do is simply go in and say, that information word is written wrongly. I'm just going to rewrite it. And that's editing. And that, I think, is the ultimate goal of the whole, if you like, the genome revolution that has taken place, which has allowed us to attribute many diseases to mutations and to know exactly what those mutations are. There's an awful lot of describing. We can describe in exquisite detail the root causes of diseases. But what we have to be able to do in the end is rewrite in order to correct those mistakes. And that genome editing, everyone was probably heard of CRISPR. That was a big breakthrough because it allows you to target a specific sequence in the genome. There's 3 billion bits of information in the genome, and to be able to just correct one of them requires an unbelievable level of specificity. That is now achievable by CRISPR. But the only thing that CRISPR does at the moment is make a cut. People are working frantically on CRISPR, but it looks as though the ability to rewrite those mistakes is not quite high enough frequency yet for us to be able to go in and correct. And the other point is that although, we were able to switch on the MECP2 gene in these mice that didn't have a functioning MECP2 gene and reverse their symptoms, what we had to do in order to do that was for every cell, or the vast majority of cells, of which there are billions in the brain, to reactivate that gene. So the efficiency with which you have to deliver the editing has to be incredibly high. All these things are challenges, particularly the delivery. We use viruses The AAV9 virus you will have heard of, perhaps. It's basically it's a virus that, like all viruses, is interested in propagating itself. It's a sort of helper virus, actually, just goes along for the ride and gets replicated when another virus goes in. But we take out its DNA and put in whatever we want, and then it becomes a vehicle that allows us to deliver in the brain. And if you inject AAV9, you can inject it into the brain, and then it progressively spreads. It doesn't seem to do anything nasty. It seems like integrate into the genome very much, and it gradually spreads and hits more and more cells. But at the end of that process, it's not clear that it hits enough cells for us to be able to make a big dent, for us to be able to correct large numbers of cells. So there are technical problems in just being able to deliver to enough cells. And there are also problems with the way in which you edit once you get there. There are incredibly ingenious tricks that allow you to do that. And one, I guess we're sort of waiting to see whether any of those tricks that get over the problems of editing actually really will work with a high enough frequency that you can correct. So it's still a tantalizing aspiration to be able to edit the brain, but it's not something that has been achieved convincingly yet, in my opinion.
So let me just make a couple of comments. So I want to make sure that our viewers pick up the nuances here. So, you know, all of our community is excited about gene therapy trials that hopefully will get back on track and start soon. That's not editing. That's what I call gene replacement. To kind of give an accurate title to it. You're not fixing the mutation that's there. You're adding extra copies of MECP2 to compensate for the mutated one. Closest to the clinic, and it will use AAV9 vectors. And while, yes, there's no way that we're going to deliver MECP2 to every cell, nor really do we want to, because about 50% of the cells have normal MECP2 already. But your work and other labs have also shown that if we can increase MECP2 a bit, we actually get improvements in symptoms.
I think this is sometimes overlooked, that you know, many of the drug therapies make hardly any difference. And it's true with a number of human disorders, not just Rett syndrome. You get a marginal increase, a marginal improvement. But the amount of improvement you get it from putting in the right amount of MECP2 is obviously, you should get a very, you know, almost back to normal. But if you put in half that amount, or even a quarter of that amount, in a mouse, you still get a huge improvement compared to the situation the mouse was in before. So there is a sort of a therapeutic window which doesn't get towards the level where you end up with too much MECP2. You don't want that either, because that also is bad. But there's a huge range from, I would say, 10% of the normal level up to 100% of the normal level. That is always, all this is desirable and may have a significant effect. It does, it certainly does in mice, but you know, this question needs to be answered yet in humans. Right. So there's a lot of scope for sort of for relatively inefficient delivery to make a difference, even though it would not qualify as a cure. That's
right. That's right. And then, you know, so gene therapy is or gene replacement, to be exact, is the closest to the clinic, and we look forward to those trials starting. A more, I guess, elegant way of addressing the problem is the editing that you were speaking about.
Yes, that's right. That was the sort of, that was the armchair wish. The gene therapy, as you say, or gene replacement, this is clearly practical, and in the mouse it's been done, and it works pretty well. The people are frantically searching for something better than AAV9, but it could be, as you suggest, that AAV9 will give us a huge advantage already. But in a mouse, there is, frustratingly, a version of the virus, it's related to AAV9, that actually infects 90% of all the cells in the brain. So this is wonderful. But it doesn't work in even other strains of mice than this one strain of mouse, and let alone in monkeys or humans that would allow these one to get it towards human. But the fact that this virus exists is incredibly encouraging. It says that if you find the right virus, and many of these viruses are literally found, they're not constructed, but now people are trying to make viruses that work better. So that it's getting much more sophisticated, and there's every reason to believe that somewhere out there it will be possible to have a virus that hits enough cells. But in the right now, one can probably make a difference without it, as Monica was saying.
Right, and with the AV9, it's the gold standard for now for delivering. And we're in constant contact with many people, many labs that are focused on finding other vectors, better vectors. And, that's news that's going to spread very quickly should that happen. And also there's another field, non-viral vectors, things like exosomes, nanoparticles. It could be that in the future there's a delivery platform that's not viral based, which has advantages, easier manufacturing and no cargo space limitations and no immune responses. So, this is a field we're keeping very close eye on, but for now, AV9 is what we have.
Yeah, RSRT is incredibly active in all these areas and is also not putting all its eggs in one basket. It's been instrumental in getting the AAV9 to clinical trials, but it's also pursuing other directions as well. I mean, I must say, I can't wait to see the results of the AAV9 gene therapy experiments.
Me too.
I shouldn't call them experiments, trials is the right word.
Me too, and I'm sure every one of the people on the phone today, parents, feel the same way. So you touched a little bit on some of the most exciting technologies. You mentioned CRISPR. Is there anything else that you want to mention? I know, for example, there's a follow-up to CRISPR called Prime, which is prime editing, which is still early days. But I think the concept that I would like to get across is that there's a lot of smart people working in the editing field, and progress is happening quickly. And although we're not there today, I think I'm very encouraged by what I see. In terms of progress, and I think we'll get there in the next few years.
I think there's that, which I totally agree with. I mean, the amount of activity in this area and the amount of creativity that is going into the ingenuity to devising new approaches is absolutely huge. There's bound to be consequences of this that will improve. And the second point is that when people are investigating these things, they're aware of Rett syndrome. Rett syndrome is on their map. It's not as though they're looking at many other things and have forgotten about Rett syndrome. Everybody knows that it's reversible because the preclinical data is incredibly valuable thing to have as a basis for moving from working on animal models to humans. That also is a huge advantage of Rett syndrome, that it's on the map, on the radar.
Yes, true. And in large part because of high-profile publications from you, from Dr. Zogby and others that have helped to put Rett on the map. And also the fact that we have this prospect of reversibility, which is really powerful and not every disease has. And so I think it makes it kind of a good go-to disease for labs and companies. So I want to leave time for Q&A. So let's wrap up this part, by asking you whether there's anything about research that you'd like families to understand, or a message that you'd like to share with families. There's a couple of hundred, parents watching you now, and all of us, myself included. We feel a great sense of urgency. Anything you'd like the community to know?
Yeah, I think science itself is a sort of a desperate attempt by human beings to understand the world. It's got a few tricks in it, like doing control experiments, for example, because humans are, not very good at putting aside their own self-interest to see the world as it really is. And what science is, and life sciences, is a series of tricks that are passed on from generation to generation as to how to make things reliable. It is actually the only way of being able to understand living things. And unfortunately, it's a bit slow and it's a bit random. The reason why I work in Rett syndrome is not because I thought from an early age that I would like to cure neurological diseases, but it's a bit of a random walk. And quite often, knowledge itself turns out to be valuable somewhere. Because there's an awful lot of ignorance, and as soon as you start to learn about things in detail, suddenly you find, and this has not only happened to me but happened to many others, that it has relevance in a specific situation. So science is messy, it's done by humans, and that automatically makes it somewhat meandering, but it's the way we get to understand things. And it's frustratingly slow for the reason I mentioned earlier, that living things are a bit, they're not like computers with bits of information. It's either 0 or it's 1, and it can't be anything else in a particular bit. They're rather squishy. They're made mostly of water. Nearly everything is nothing is really off, or it's always, any switch is a little bit on. This makes it difficult, and also you get redundancy. So not only is a certain process done in one way, but it's done in another way at the same time, in case the first way fails. That makes it difficult to study, because if you take away the first way, there's still the second way. So you can't tell there's anything wrong always. I know this sounds very abstract and philosophical, but all I'm really saying is that biological systems are a bit tricky to understand, but actually, given the new tools we have, like the sequencing tools, mass spectrometry, all these other techniques, there's never been a better time to understand biological systems, including the brain. And I think, as I mentioned, Rett syndrome is in a prime position to be a beneficiary of this. And I just really hope that it's quick enough to benefit the people who are listening now and the individuals they care for.
Thank you. Okay, I'm going to start reading off some of the questions. So this one is from Matthew. He says, then do you believe we have to first understand each and every one of MECP2's actions or roles in the body to be able to successfully treat Rett syndrome?
Well, if the answer to that is, it depends which route you're going, but the answer is not necessarily. A thing like penicillin was used way before anybody understood how it worked. And actually what the reversal experiment says is, if you put back the gene, which we do know is involved, then the brain gets better. And you don't need to know everything that MECP2 does in order for that to happen. So that all the editing, gene therapy, protein delivery, whatever methods do not require you to know absolutely how MECP2 works. If, on the other hand, you're going to devise drugs, pharmacological ways of treating, then you need to know, yes, MECP2, when it's missing, leads to this downstream effect. And there's a target there that I can challenge. There is, for example, too much of something, and I can find a drug that inhibits that thing, so that even though there's too much of it, leading to a bad effect in the brain, I can inhibit it. So that sort of route remains, still requires you to understand how the protein works. And it is just possible that one will need both approaches, that actually, combinations of drugs are more and more used, and it could be that that will continue to be the case. I would say, as a scientist, it's better to, if you're trying to fix something, if you understand how it works. but with the qualification that if you do gene therapy and rewrite the genome, then you don't necessarily need to. Messy answer, but hope you got some of it.
Okay. Next question comes from Mustafa. He says, do we know which are the downstream genes impacted by MECP2 that causes Rett?
The reason why it's taken a while to actually understand how MECP2 works is that it affects hundreds, if not thousands of genes. It's a kind of global modulator, and it doesn't switch genes off. It simply lowers their expression in a way that depends on how much of this methyl mark they've got. So when you take it away, you get, instead of it being, let's say, an arrangement like this, you sort of get a little bit like this. And we think, we're pretty sure that that is the reason for the problem. Now, your question is still valid because it could be that many of those changes don't matter and that there are a few that do. And actually, that's something we're working on now in my lab, to try to find genes that are misregulated, that go awry among all the many hundreds or thousands that change, that actually matter more than others. And we've got a smaller set of genes that we're focusing on because they keep coming up over and over again in different studies we've done. We don't know if they really are more important than the others, but if they are, they would be potential targets, drug targets.
Okay, great. And Mustafa has a follow-up question. Is there a risk if the gene replacement, MECP2, is delivered to a healthy cell?
Yeah, well, we haven't really mentioned that, but duplication of the MECP2 gene looks as though it is bad. So it causes MECP2 duplication syndrome. So one wants to avoid too much. And as Monica said, and as you allude to, half of the cells in the brain of a child with Rett syndrome, a girl with Rett syndrome, have the right amount, and half of them have very little or none. It's the ones with very little or none that we're trying to get to, but in the process, we can't distinguish them. We can't say, only do gene therapy in those cells. So you need to get into this, you will automatically go into cells that already have enough and give them more. So there is a risk that causes the very problems that you get with duplication syndrome. The only thing I would say about that is that the work that we have done has suggested that there's an element of flexibility upwards, if you like, in the amount that you can put in a mouse system. So in other words, too much, a little bit too much is not that much of a problem. And I would say that's been mimicked by the gene therapy experiments in mice, where you're doubling the amount in certain cells, half the cells, and putting it back in the other half, and nevertheless the effects are not that bad. It's something one has to be aware of. And the reason why I stress that a little bit is going to be good is that if you put in a little bit and get the benefit, you're not actually adding much to the cells that already have the right amount.
I'll add one comment here too. So with the duplication syndrome, it's a section of the X chromosome that's duplicated that always includes MECP2, but it also includes other genes as well. It's not only MECP2. For example, IRAK1 is always included in the duplication syndrome. And IRAK1 is involved in immune issues, and duplication kids have a lot of infections, respiratory infections. That could be due to the IRAK1, not necessarily the MECP2 being duplicated. So I think that's something to be aware of. And then the other comment is that, children with the duplication syndrome had the alteration, the duplication from conception through development onward. It might be a different situation when you're delivering extra copies of MECP2 into a cell that's already developed versus one that has to go through the development. We don't know for sure, but it, it could be less severe. Those are just two observations.
Yeah, we know somewhat less about the basis of duplication syndrome than the basis of Rett syndrome. As Monica mentioned, there are other genes. But we don't know that it's safe to have too much MECP2
Yes.
either, except from these experiments I discussed in the mouse where it looks to be relatively tolerant of too much.
So there's two questions about MECP2 levels. I'll ask the first one from Amanda. Is there a way to measure how much MECP2 an individual has? She says, for example, my daughter seems to have a less severe Rett, even though she has a classic mutation.
Yeah, this is something that's difficult. Obviously, you can't do biopsies in the brain, so people tend to measure MECP2 in the blood, even which is not a relevant tissue as far as the symptoms are concerned. I think there isn't an ideal way of measuring MECP2 abundance in a living individual. There are, however, reasons why one might have a mutation that causes classical Rett syndrome that does not appear to manifest itself severely. And one obvious possibility is that X inactivation isn't always 50-50. So the situation, just to mention it, there are two X chromosomes. One of them has the mutation in it, in the MECP2 gene, and the other it doesn't. It's a normal, unaffected chromosome. And what female cells do is early in development switch off one of those X chromosomes. And they either switch off so it's random in many individuals. So half of the cells have switched off the mutant X chromosome. So that means that they're functionally normal with respect to MECP2, if I can use the word normal. And the other half have switched off the normal gene, and therefore the only MECP2 gene they have is the mutant one. And so there's this salt and pepper mixture in the brain of cells that are normal and cells that are abnormal with respect to MECP2. So in many individuals, that's a 50-50 switch, random chance. But actually, in a significant fraction of the population, way more than 1 in 100, so then there is a skewing. And one of the X chromosomes is preferentially inactivated. So if you preferentially inactivate the chromosome that has the mutation on it, then more of your cells have perfectly normal functional MECP2. And that means that you have a higher proportion of normal cells in your brain, and that's going to make you, on average, less severely affected. And that you can model that in the animals. So it's pretty clear that's the case. So the suspicion is that is a component of the reason why Rett syndrome is so variable, even though a mutation in one person is very severe, and in the same mutation in another person is not. And that's a potential explanation for that. But we would like a better way of measuring how much MECP2 there is in the brain. We really would, but it's difficult to think of a way of doing it non-invasively.
Yeah. And a question kind of along the same lines from Mark. Is appropriate amount of MECP2 likely dependent on the age of a Rett patient?
That's an interesting question. I mean, in the mouse, what happens, and in the human, MECP2 goes up in amount in the brain. So in a newborn, the neurons, the nerve cells are all there, but they have relatively little MECP2. And then during the subsequent months, they get more and more and more. And it keeps on going up a little bit, but it does plateau during infancy. The assumption is that once you've got the right amount, then that's the amount you need throughout your life. My guess is that that's probably correct, and you don't need different amounts of MECP2 at different ages. That's my guess. But if you just think of what you would need to do in a human in order, given that I've said we can't really measure accurately in a living brain how much there is, you'd really need to do an enormously elaborate experiment that would take a very long time to do to be able to answer that question properly. I mean, I think it's a reasonable assumption that you need one amount, and once you've got there, you don't need more. But of course, no protein is so stable that you just make it and then leave it. All proteins have a half-life. They decay. And so you constantly have to replenish. And that's an ongoing thing throughout life. You need to make MECP2 to replace the MECP2 that's gone off, if you like.
So D asks a question: how does the vector know which cells to target? How does the vector discriminate between good cells and cells that need replacement? So
yeah, that's the problem. It doesn't. It can't distinguish those two. And so you add more to cells that don't need it. And that's, sorry, I didn't make that clear before. That's, there are cells, the cells that already have enough are going to get more. And what one really needs to know is whether that matters or not. And it's going to be half the cells. It probably will matter a bit if you get to very high levels, but if you stay low, it might not matter very much. I think one could argue that quite persuasively at the moment.
And Dee, that's why we mentioned earlier on that an editing approach, an approach that actually gets in there and fixes the mutation, whether it's in DNA or RNA, or even reactivating the silent healthy MECP2, are approaches that are in a sense more elegant because it bypasses the problem of potential overexpression.
But people are also trying clever solutions to this. So for example, your central heating stays at the same temperature because if it gets too hot, something comes in and switches it off and says this temperature is too high. So if you had something like that, a feedback mechanism for MECP2, and people are trying to think of ways of getting that, so that if a cell suddenly starts to have too much MECP2, it says, whoa, slams on the brakes and stops making it. So it would be a regulated system rather than just chuck it in and hope for the best.
Yes, sure. And we're funding a number of those approaches. And I think that's a situation that's going to prove, so that would have to be built into a gene therapy construct, just to be clear. A mechanism that regulates levels of MECP2. So there's a comment here also from Dee that I thought was interesting. She says, maybe the COVID-19 virus could be used as a delivery since it infects such a variety of cells. I don't know how well it gets into the brain. I mean, people are having some seizures and some neurological issues, but I don't know whether it's a vector that preferentially gets into the brain, which is really what we want.
I don't think it seems to at the moment, but if you were to inject it, I mean, as I understood it, it goes for respiratory epithelium. Epithelium is just one of those long words that means skin, if you like, the sort of skin on the surface of your lungs and the tubes that lead into the lungs. That's what it likes, because the receptor that it binds to, that it's picked out as being its way into the cell, is expressed at a high level there. I don't know if you could hijack it. It would certainly teach it a lesson, but it's also an RNA virus, and it's a very long RNA. It's 30,000 nucleotides long, and that's pretty big to engineer. But I mean, that's the sort of thinking that goes on. Viruses are potential vehicles, and you can bet someone's been thinking of that sort of thing. Maybe you can't bet it. Maybe you're the first person, so you should cash in on this idea. Yeah.
So we're at the hour mark, so I want to wrap it up. But for people who ask questions that we weren't able to get to, we'll email you our responses. And if I don't know the response, I'll reach out to you, Adrian. So thank you so much for joining us, and thank you to all the viewers who participated and asked questions and registered. We hope that you're managing through this difficult time, and that your daughters and sons are healthy and remain healthy. And for Professor Bird, I just want to thank you and your many lab members over the years for your contributions.
I'd like to thank my lab members as well, because I've spent a lot of time in my office, and they're actually at the coal face doing the experiments. So I've been very lucky with who I've had working with me.
And we wish you Godspeed moving forward, and you know, a lot of hope for continued progress.
Here's hoping.
Thank you so much.
Bye.